Our keynote speaker is Dr. Richard Losick, who is the first recipient of MSI's Distinguished Achievement Award and will be giving our keynote. Now he's going to be introduced by Dr. Niels Bradshaw, who is an assistant professor of biochemistry at Brandeis University, where he and his group study the regulation of protein phosphatases and the evolution of cellular signaling. Now, Niels did postdoctoral research with Professor Losick, and when I asked him if he would be kind enough to introduce Rich, he responded with great enthusiasm. And I imagine that Niels has many wonderful things to share about Professor Losick and the impact he's had on microbiology and on many scientific careers. So Niels, if I may turn it over to you.
So, of course, it's with great enthusiasm that I'm looking forward to hearing from my science hero, my mentor, my friend, and now my collaborator, Rich Losick, for the Distinguished Achievement Lecture. It's also an unusual thing because Rich has been many of those things to many people in this room, some much longer than me, and he's also had a singularly important impact on life sciences research for longer than I've been alive. And so I thought that what I would do is just highlight, for people who don't know Rich as well, a few of the sort of like earth-shattering discoveries that he made. And then I'd like to offer a hypothesis for why Rich has had such an important impact on science. So when Rich first came to, one of Rich's first big discoveries came after after he'd been a junior fellow at Harvard. He and Jan Perrow had a joint lab. This was in the heyday of molecular biology, and one of the structure of DNA was known. It was known that it was transcribed into RNA, and that's what made protein. But it wasn't known how RNA polymerase transcribed particular genes at particular times, which was necessary for cells to survive in different environments or to differentiate into different cell types. And so they had hypothesized that there was a sigma subunit of RNA polymerase that was only needed for the initiation step, and that there might be alternative sigma factors that carried this out. So the first thing that they discovered was the existence of these transcription factors. So this is a picture of a gel from Jan's notebook in 1974, where they found two bacillus phage sigma factors that are here on this gel. And they could then show later that these different sigma factors were responsible for transcribing specific subsets of phage genes. This gel was particularly important to me. Rich asked me to help him scan it for an article that he wrote in Cell about sort of this discovery. And it was sort of amazing to me to get to hold this gel, because these were the very molecules of transcription, of the first transcription factor that may have ever been knowingly seen by anybody. And I just wanted to show quickly, I took a picture of Rich unpacking this gel from the notebook that had been stored in their garage. So anybody who has to put together a new data management policy for federal funding, this could maybe be a template for your efforts. So for these discoveries, in 2007, Rich was awarded the Waksman Prize from the American Society of Microbiology for the discovery of alternative sigma factors. And he'd actually discovered in his lab and with Jan multiple other transcription factors. And so there are two things I want to point out here. So one is that what Rich could have done is, this was sort of the first transcription factors, gone and tried to figure out how eukaryotic transcription works. And instead they left this to Bob Tejan. So you'll see in the bottom right-hand corner, Tejan's RNA polymerase was used as a marker on this gel. And Rich makes a very convincing argument that T should have won the Nobel Prize for the work that he did following up on that. But what Rich did was he went back to Bacillus subtilis. And so he asked, they'd originally worked on Bacillus subtilis because it did the kinds of amazing transformations that Amy just talked about. And so Rich wanted to work that out. And so, and he did. So this is an image of some of what they found out. So that it turned out that there were a bunch of different cell-specific sigma factors that controlled the developmental program. Rich in his lab figured out how they talked to each other, how they were initiated. And I purposely stole this figure from a review article that was written by Bob Horwitz and Ira Herskowitz. That was in the early days of trying to understand developmental biology, and this work that Rich did was really influential for everybody understanding development. It wasn't microbial development, this was just how do cells differentiate. And in 2009, he and his longtime colleague, friend, and collaborator, Lucy Shapiro, were awarded the Gairdner Award for their discovery of the mechanisms of asymmetry and how it was, which was really important for cell growth. And I thought that this was particularly important, again, because they were just awarded it for the discovery of asymmetry, not microbial asymmetry. This was a very general discovery. So what did Rich do? He went again back to B. subtilis and wanted to understand the molecular mechanisms of those kinds of transformations. And in the process, discovered fundamental things, for example, about the mechanisms of cell division and other things. And then, so for example, this is some electron micrographs done by Adam Drix, looking at the compartmentalization of transcription factors. Rich was one of the, I think, was the first person to use a GFP fusion protein in bacterial cells. And so this work was also recognized in 2012 with the Horwitz Prize from Columbia University, along with, again, Lucy Shapiro and Joe Lichtenhaus for the discovery of the internal structure of bacterial cells. So anyway, really remarkable, 3 major awards, all for different things that Rich discovered. The thing that I want to do next is to sort of explore a hypothesis for why did Rich make such a big impact and how did this happen? And so the hypothesis that I want to propose, Rich tells a story, and you can read a really lovely review that he wrote in the, I think it was in the Journal of Biological Chemistry. He calls it his love affair with B. subtilis, where he talks about how wonderful it was to study B. subtilis. But the hypothesis I want to propose is that Rich really emulated B. subtilis, and that that's the secret for his success. That it wasn't just that he was a keen observer, but that he adapted those things into his own work. So here's my hypothesis that Rich is like B. subtilis, and I'm borrowing some of this from a primer that Rich wrote that I think is a wonderful thing to read in Current Biology recently about the things of B. subtilis. And so in this, what he explores is how is it that Bacillus subtilis is able to adapt and respond to varied environments. Rich says that it's versatile, and there are 3 specific traits that B. subtilis has. And so the first of these is that B. subtilis has specialized cell types. We've already talked about this sporulation. Amy gave a wonderful introduction to this. So what are Rich's specialized cell types? So one, we know he's a scientist and an incredibly versatile one. Second, Rich is a teacher. And his teaching has been nearly as renowned as his science. He, for many years, taught advanced introductory molecular biology, but then went on to teach, develop it with Dan Kahn and Susan Mango, a class on Life Sciences 1A that merged general chemistry and introductory molecular biology. I used the electronic textbook that they taught in my advanced biochemistry classes. It's an amazing resource. Since then, he's gone on now to also teach science communication, a class that was so popular that they're still giving classes after the end of the talks after the end of the semester, and also a class on the history of molecular biology, which is the basis for some of what he's going to talk about in this class. Rich also had 2 cycles of an HHMI-funded professor program where he brought students into research labs. And I can't even begin to recount all of the things that he's done as a teacher. He's also been an editor. So Rich has had a huge impact on the research that we all read. He was on the editorial boards of Science and Cell, and was one of the founding senior editors at the journal eLife, and had a huge impact on those publications. He's also a colleague, and much to Rich's embarrassment, every year there's an award that's given. I can see you're already crawling under your seat a little bit. There's an award given in the department, the Losick Award for Departmental Service, that's a reflection of the dramatic impact that Rich has had on the institution. Okay, so what else does Bacillus subtilis do? Make specialized cell types. It can also be motility or lack thereof, is what Rich cited. So in this particular image here, it's looking at how Bacillus subtilis can either exist in a motile or a non-motile chain form. This work on this is also part of a collaboration with Johan Paulsen over at Harvard Medical School that looked at stochasticity and the role of this. So now if we think about Rich in this perspective, Rich is definitely in the lack thereof department. He's been on the 3rd floor of the biolabs, again, for longer than I've been alive. And that 3rd floor of the biolabs is a remarkable environment. Rich has a piece on the MCB departmental website about the 9 Nobel Prizes that were sort of incubated in that environment and in that space, and the many more that could have been awarded. I think that Rich likes to see it as that this was a magical environment that he got to be a part of, but I can't help but think that he must have also been a large part of what that environment was. Anyway, so Rich stayed put. But that's not the whole story, because Rich has also trained a huge number of people that have gone on to have a huge impact in science all over the world. And so I think, again, like subtilis, having both abilities has been really special. Here's a picture just from 10 years ago. We celebrated Rich's 70th birthday, where many of these people came and gathered. And this year we're going to celebrate his 70th birthday again. Okay, so what else does Bacillus subtilis do? Bacillus subtilis also has multicellularity. So Rich worked out the mechanisms of this through a multi-decades-long collaboration with Roberto Coulter that led to just like an absolutely stunning array of work and a new way of thinking about bacterial communities. And so how does this reflected in Rich's own life? So one is through the many collaborations that I've already mentioned throughout this, including some that haven't come up yet, including later stages of his career, like initially branching out to work on Staph aureus through a program initiated by Mike Gilmore, and then finally with collaborations with Dan Kahn and Suzanne Walker and Staph aureus. The other way is through the MSI itself. So the sort of one of the origin stories of the MSI was that the Harvard had an initiative to create interdisciplinary centers. And on the way back from an administrative meeting that Rich was at with us, walking with Dan Schrag and Colleen Kavanaugh, they hatched the idea of having a multidisciplinary microbial thing. So now, as Rich tells it, at least in the brilliant wisdom of Harvard administration, that idea never came to fruition. But the idea of having an interdisciplinary microbial collaboration withstood that, and they decided to do it anyway. And although Rich was never a director of the center, he's had a huge impact on that. I think even in some ways that reflect his obsession with Bacillus subtilis. So if any of you had ever been to the Chalk Talks, which were hugely popular events, Rich insisted on having them in a small room so that we were all very close together, because he thought the communication worked best when you were rubbing elbows with your neighbors. Okay, so in terms of my hypothesis, I think I've shown you a strong correlation that Rich is a lot like Bacillus subtilis, and that may be one of the explanations for his work. But I don't have any, so where is there a causal relationship that I can relate to that? So I have one piece of data that I want to present. So when I first joined Rich's lab, he was actually in the process of acquiring a whole new wardrobe because he had just been to China and learned about how studies of the traditional Chinese diet affected the microbial communities and created much more healthy microbial communities, which of course are highly enriched in firmicutes and other organisms related to B. subtilis. And so Rich redirected his whole diet based on this, not because of the effect on him, but because of the effect on his microbes. And so with that, I think that we can conclude that one of the secrets to success is that you ought to not just study your microbes, but be your microbes. And so with that, I'd like to ask that you offer a short but raucous round of applause, because I can't wait to hear what Rich has to say. Thank you.
Okay, great. I'm kind of speechless. Okay. I'm going to change gears and ask you a question. What do you think was the greatest discovery in biology in the 20th century? What was the greatest discovery in biology in the 20th century? I need to carry this with me if I'm going to walk around and hand this. Anybody just venture a guess? What? This? So, what do you think? What happened? You're all too young? Is that the problem? You know what happened? What? Antibiotics. Well, that's not unreasonable, Steve. But I'm going to suggest that the greatest discovery was that the genetic material is DNA. And but DNA is the genetic material. Of course, antibiotics are hugely important in humanity. And we just heard a great talk over on your side of the river by about the history of this. I want to tell you the story of how this was uncovered. And this is a story in 3 parts. First, I'm going to tell you a prequel that takes us back to 1928 and Frederick Griffith, who discovered something that he coined as the transforming principle. Then I'll come to the main story. In 1943, Oswald Avery and his team of Colin MacLeod and Macklin McCarty purified Griffith's transforming principle, and they showed that it's DNA. But they faced skepticism, and it wasn't widely accepted. And why was that? And then there's a sequel to this story that took place in 1952 by Al Hershey and Martha Chase. With less convincing evidence, they gained widespread acceptance that DNA is the genetic material. So by way of background, and luckily, we've already from Jackie and Anne, the star of the story, strep pneumo, has already been introduced today, but let me tell you a few more things about it. So Griffith and Avery, their discoveries are based on Streptococcus pneumoniae, a big star today and a star in this history, which of course causes pneumonia. Virulent pneumococcus strains form smooth-looking colonies on agar plates, but there are mutants called rough mutants that make rough-looking colonies on plates. So here's a rough mutant, and these are smooth mutants. These are virulent, and these are avirulent. The virulent cells and smooth colonies are surrounded by a polysaccharide capsule, whereas the avirulent cells in rough colonies lack the capsule. So here is the virulent cell, and you can see this thick polysaccharide capsule that surrounds it. Different virulent strains produce chemically distinct capsules that can be distinguished serologically. So there's serotype 1, serotype 2, serotype 3, and so forth. Okay, that's the background. Now, Griffith. So he was a medical officer of the Ministry of Health in London. He published this impactful paper, The Significance of Pneumococcal Types, in 1928 in the Journal of Hygiene. And he discovered that heat-killed virulent pneumococcus released a transforming principle that converted avirulent pneumococcus to virulence. Sadly, he died in 1941 during a German bombing raid on London, so he didn't live long enough to see the fruits of his discovery, which unfolded just 2 years later. Okay, I have a cartoon that summarizes the essence of what Avery found. If he took a mouse and he injected it with the rough mutant, R cells, rough mutant cells, the mouse lived and you could recover R cells from the healthy mouse. If you injected the mouse with the smooth virulent cells, the mouse croaked and you could recover the smooth virulent cells from the mouse. If you heat killed the S cells, the mouse lived. Of course, the bacteria were killed and no cells from the mouse. Now, here was his impactful discovery. If he mixed the heat-killed cells with the rough cells and injected that mixture into the mouse died. And from the mouse, you could recover S cells, which hadn't been there to begin with. Somehow, something from the heat-killed cells was converting the rough cells into the virulent S cells. Not only could this transforming principle convert a rough mutant to a smooth capsule-producing strain, but it could also transform the rough mutant to a new serotype. Let me illustrate that for you. Thus, a rough mutant derived from serotype strain 1 could be transformed to serotype 2. So if we start with serotype 1 and we derive a rough mutant from it, and then transform with heat-killed type 2 cells, we can recover serotype 2. Not only are we going back to virulence from rough to smooth, but to a new serotype, okay? That was Griffith's discovery. Okay, now the main story. The main story, the main story is Avery, McLeod, and McCarty. They made their transformative discovery in 1943. They published their findings the following year in the Journal of Experimental Medicine. Here he is, Oswald Avery. He worked at the Rockefeller Institute in New York City, now called Rockefeller University. And he was joined by McLeod and Mac McCarty. Their mission was to determine the chemical nature of Griffith's transforming principle. Well, importantly at this point, it was known that transformation could be carried out in a test tube. You didn't need to have mice. Under the right conditions, simply mixing heat-killed cells with our bacteria, and you could get this transformation. This audience will appreciate that this was what we now call competence, and certain kinds of bacteria, especially gram-positive bacteria, have genes that under the right conditions will allow cells to be transformed by something that we're coming to. Okay, and here's Avery's team. Here's Colin McLeod, and here's Mac McCarty over here. Mac lived well into his 90s, and that's important for a little tidbit that I'll come to later. So how do they purify the transforming principle? Actually, most of these experiments were done by Mac, and that'll be important. So they prepared a cell extract of pneumococcus. They treated with chloroform, which precipitated the protein. They treated with an enzyme that degraded capsule. They treated with ethanol, which precipitated DNA as fibers. Now, at this point, a lot was known about the chemistry of DNA, and a man named Alloway had shown that ethanol treatment would cause DNA to precipitate as fibers. As you can see, here's a glass rod with DNA fibers on it generated by ethanol treatment. They took those fibers and they dissolved them in saline solution, and they repeated the precipitation, solubilization multiple times to increase purity, and they did so without losing activity. Okay, what is the argument that this transforming principle is DNA? First of all, the material was fibrous and viscous like DNA, as I've said. Elemental chemical analysis after complete combustion carried out at Rockefeller was compatible with DNA, but not with protein. DNA is rich in phosphorus. Protein has few, if any, phosphates on it, unless it's phosphorylated. And here's an experiment. Here's a table from their paper. So in theory, DNA should have a nitrogen to phosphorus ratio of 1.69, as highlighted down here. And here are the results of 4 different preparations of transforming principle. And you can see that they closely approximate the theoretical value. If it was protein, the ratio would be much, much higher than this. The transforming activity was insensitive to trypsin and chymotrypsin, RNase, and the polysaccharide degrading enzyme, but it was destroyed by DNase. Now let me give you 2 quotes from what Avery and his buddies had to say about all this. It is evident, therefore, that not only is the capsular material reproduced in successive generations, but the primary factor which controls the occurrence and specificity of capsular development is also reduplicated in daughter cells. The induced changes are not temporary but permanent. That's my bolding to emphasize that sounds like hereditary. changes, doesn't it? Reduplicated and permanent. If the results of the present study on the chemical nature of the transforming principle are confirmed, then nucleic acids must be regarded as possessing biological activity. Despite this, there were some who felt, and having to do with reasons about its personality that I'll come to, there were some who questioned whether Avery really appreciated the significance of his own work. Did he really understand the impact of what he had done? And that clearly is the case, and that's revealed in 1943 in a letter that he wrote to his very dear brother. He was very close to his brother, Roy, who lived in Tennessee, and later Avery would join him he would retire. Avery never was married, and he moved to Tennessee after he retired and to live with his brother. So this is in his letter to his brother. Who could have guessed it? If we are right, and of course that is not yet proven, then it means that nucleic acids are not merely structurally important, but functionally active substances in determining the biochemical activities and specific characteristics of cells, and that by means of a known chemical substance, it is possible to induce predictable and hereditary changes in cells. This is something that has long been the dream of geneticists. Sounds like a virus, maybe a gene. He got it. Okay, and just to convey his sweet nature, this is the end of his letter. This is a photograph of it. Forgive this rambling epistle. With it goes my love and thought in hope of better things ahead. With heaps and heaps of love, affectionately and faithfully, OTA. Dear man. Well, all these experiments were done with capsule formation and virulence. Was this really general? Well, Avery was joined by a postdoc named Harriet Taylor, who carried the experiments further, and she carried out transformation for streptomycin resistance, nothing to do with virulence, but antibiotic resistance. Now, Harriet Taylor, I just pause for a moment. She married a man named Boris Afrusi, a very prominent scientist in his own right, and she's the mother of Anna Afrusi, who was an undergraduate here, who I knew, and now runs a big division at the Embo Labs in Heidelberg, and whom I've had discussions about all this. But sadly, Harriet Taylor, who you're about to meet, died from cancer very young. So it's really tragic, because imagined quite a career for her. But she appreciated what Avery was doing, joined his lab, and carried the work further. And there is this educational video from way, way back that I'm going to share you. You will see her. It's not her voice, but it's her in the video.
We have discovered that some bacteria become resistant to drugs, to antibiotics. This resistance is hereditary. In other words, the bacteria have acquired a mutation for resistance. This is how we can demonstrate what happens. Both of these plates contain an antibiotic that kills bacteria. This dish has been spread with ordinary pneumococcus bacteria, the kind that causes pneumonia. None of the cells here have grown because the antibiotic has killed them. The second dish has been spread with the same kind of bacteria, except that this strain is resistant. These cells are flourishing. Now we can extract the DNA from the resistant bacteria, and we get these pure fibers of DNA. Here you can see the fibers more clearly. We have put a little of this DNA into a tube. Now we add to it the bacteria which are not resistant and let them grow for a little while. Finally, we spread these treated bacteria from the tube on a dish containing the antibiotic, spelling out the letters D, N, A, and leave it in a warm place for a day. By the next day, something surprising has happened. The bacteria have multiplied. Their heredity has been changed.
Research on DNA is one of the great new...
Okay. I can't top that. So every great story has to have a villain, right? We have a villain where? At the Rockefeller. His name was Alfred Mirsky. Mirsky was prominently known as a protein chemist. He was obsessed with protein, very prominent protein chemist. And he argued that there can be little doubt in the mind of anyone who has prepared nucleic acid that traces of protein probably remain in even the best preparations, and that as much as 1 or 2% of protein could be present in a preparation of pure protein-free nucleic acid. Well, in response to that, someone else who teamed up with Avery by the name of Roland Hotchkiss, a very prominent scientist in his own right, he further purified the transforming principle and reduced the level of protein to less than 0.02%. Now, I'm going to quote now my friend Greg Petsko, now at Brigham and Women's Hospital, knew Mac McCarty at Rockefeller and wrote obituary, and he has some very beautiful things to say about all this that I want to share with you. The man who did the greatest experiment in the history of biology with his own hands and never won the Nobel Prize, even though he lived on for more than half a century after the experiment changed biology forever. His name was Macklin McCarty, and he was the junior member of the team of Avery, McLeod, and McCarty that proved that genes are molecules of DNA. It was Mac who did a lot of the experiments I described for you with his own hands. Despite the powerful evidence in the paper, this conclusion was not accepted by many influential scientists. Chief among these was Avery's Rockefeller colleague, Alfred Mirsky, a biochemist who lobbied for years to deny Avery and his team the recognition their work deserved. Mirsky was convinced that proteins had to comprise the genetic material and believed Avery's DNA samples had to be contaminated with them. He seems to have persuaded the Nobel Committee because although Avery and his associates were nominated repeatedly in the years following their discovery, they never won the Nobel Prize. Avery died in 1955, age 76. To be fair, Avery himself did not help matters. He had a quiet, self-effacing personality, presented his work in a low-key manner, and was averse to any sort of speculation. His presentations were few, and when invited to speak at international meetings, he usually sent his younger collaborators. Okay, so we're ready for the sequel. And this is the paper of Al Hershey and Martha Chase on the dependent functions of viral protein and nucleic acid in the growth of bacteriophage, published in 1952, 8 years after the Avery publication, 9 years after the transformative results were obtained. Martha Chase was Hershey's research associate. She later got a PhD and then had a very sad life that I won't get into. It was very sad. Okay, so together with Max Delbruck and Salvador Luria, Hershey was a member of the highly influential phage group. Many here will know that in the history of molecular biology, it was launched by the phage group. And Frank Stahl of Messelson-Stahl fame said of the members of the phage group, the phage church, as he was referring to it here, was led by the trinity of Delbruck, Luria, and Hershey. Delbruck's status as a founder and his ex cathedra manner made him the Pope. And of course, and Luria was the hardworking, socially sensitive priest confessor, Jewish priest confessor, And Al Hershey was the saint. Well, I'm going to push back a little bit about sainthood for Hershey, as I'll come to. So here's the famous experiment, which was carried out with phage T2, with the idea that there was a protein coat. Internal to the protein coat was the DNA, and it was the DNA that was injected into the cell. So if DNA is the genetic material, then following infection, only DNA should enter cells. So they set out to test this. They prepared phage that were labeled with P32 for DNA or with S35 for protein. After infection, the coat was stripped off the cells with a blender, a wearing blender. And then radioactive phage from the infection were collected and then used to infect fresh cells in a second round of infection. And they collected those phage, and what they found was, here was the result, 1% of the S35, but 30% of the P32 were transferred to progeny phage. And here's what Hershey and Chase had to say about that. We have concluded above that the bulk of the sulfur-containing protein of the resulting phage particle takes no part in the multiplication of phage, and in fact does not enter the cell. It follows that little or no sulfur should be transferred from parental phage to progeny. The experiments described below show that this expectation is correct and that the maximal transfer is of the order of, guess what, 1%. Well, how does 1% compare to the purity of Avery's transforming principle? I ask you. Not only that, this was shocking to me when I realized it. Not once in the Hershey-Chase paper do they cite the Avery paper. They make no mention of Avery, McLeod, and McCarty. Here are all the references, and not once is Avery present. So Hershey went on to win a Nobel Prize with Luria and Delbruck, while Avery et al. Went unrecognized. Salvador Luria, don't misunderstand, he's my hero. I knew him well, but in my days at MIT. Okay, and Delbruck, I actually met briefly. It's a amusing story that I don't have, you can ask me afterwards, but I did get to meet him. I never met Al Hershey, but Delbruck and Luria, wow. Okay, so let me ask the question, why did the concept that DNA is the genetic material not gain wide acceptance until 8 years after Avery, McLeod, and McCarty? And I have a couple of reasons that I will, First of all, there's a very influential book that was published in this era by the famous physicist Alfred Schrödinger, What is Life? And he's the person who helped draw a lot of physicists and others into the question of what makes something alive. But he believed that it was protein, probably a large protein molecule. And of course, proteins are complex. They're composed of 20 amino acids, and DNA is composed of only 4 nucleotides. So how could it encode information? And then there's the case of Phoebus-Levene. I suspect there's almost no one in this room who knows who Phoebus-Levene is. You know who Phoebus? Oh, okay, all right. So he was also at Rockefeller. All the good guys and the bad guys were in Long Island. Had reported that DNA was a monotonous tetranucleotide. Let me tell you, Phoebus-Levene was a pioneering DNA chemist. He wasn't a biologist. He figured out the 5' 3' phosphodiester linkages that hold DNA together. And he assumed that DNA would just be an ordinary repeat of AGCT. This was the tetranucleotide hypothesis, that DNA was a boring repeat of the 4 bases. Now, that had a big impact in convincing people that DNA couldn't play a role in genetics. Let me say as an aside that Erwin Chargaff, late in the decade, that overturned the tetranucleotide hypothesis and pretty much buried Phoebus Levine in the process. If I had more time, I could give a whole lecture on Erwin Chargaff. What a character he was. Endless stories about him. But he was merciless in what he did to Phoebus Levine. So even though Phoebus Levine was this pioneering chemist in DNA, very few people other than Roberto Coulter has heard of him. Did I tell you about him?
No, but you know what? It's fascinating because earlier in the day,
Yeah.
because I knew it was coming up.
Oh, you cheated.
I was just, it is a fascinating story.
Yes, okay, well, it got down. Okay, so hence the transforming principle must have been contaminated with protein. DNA was boring and not too complex, even though it was protease insensitive and highly purified. So must have been contaminated with protein as argued by Mirsky, despite all these other experiments that Avery contributed. Learning of Avery's results, the father of molecular biology, Max Delbruck, said, "You really did not know what to do with it." He later said, "It just meant that genetic specificity was carried by some goddamn other macromolecule instead of proteins." Well, even that, he didn't even see it as important. And finally, Salva. Lurius is quoted in The Eighth Day of Creation saying, the famous book on the history of molecular biology, people like Delbruck and myself, not only were we not thinking biochemically, but we were somehow and probably partly unconsciously reacting negative to biochemistry and biochemists as such. Okay, so let me pull this all together for you. Why Avery didn't get the recognition he deserved. To me, the phage group and the nucleic acid biochemists lived in different intellectual worlds. And the discovery of Avery et al. Was simply ahead of its time and largely went unappreciated. We had the tetranucleotide hypothesis that was clouding everything, and that hadn't been disproved yet. Nevertheless, and looking back, we can say it represents one of the greatest of all discoveries in the biological sciences in the last century. So let me close by saying that today we celebrate the 20th anniversary of the Microbial Sciences Initiative. And wow, this has been, what a wonderful symposium today has been. So that 20 years, 2003. Let's also celebrate the 80th anniversary of Oswald Avery's transformative discovery in 1943. And if you'll permit me, of vastly less significance, yours truly is celebrating my 80th year on this planet. I was born the same year as the Avery, McLeod, and McCarthy experiment. But to be honest, I was too young to appreciate it. Thank you all. Avery and pneumococcus. We have to raise them.
Don't go too far, Rich.
Okay.
Mike, why don't you join us up here? I have a few things I want to share. My goodness, it was a great talk. I want to thank you, Rich, not only for the wonderful talk, but for your many contributions to microbiology. I think we could have done a whole symposium on the work that Rich, his students, and colleagues have done throughout the years. I think equally important, though, is the support and commitment that Rich has shown to those who have come through his lab, and those of us who are lucky enough to count ourselves as among his peers and colleagues. I happen to inhabit the third floor, and Rich is always reminding me of all the great things that happen. No pressure, right? And on most days I'm like, I think I'm pulling down the average. But Rich is always incredibly supportive. I want to share a couple of quick stories. When I first joined Harvard, I remember reading all this cool work that had come out of Rich's lab, and I remember thinking, God, it'd be so cool to work with Rich. I also remember bumping to Rich in the parking garage in his sporty little Porsche, and I was thinking, like, wouldn't it be cool to go on a drive with Rich and talk about science? And I said, that's a little forward of me. But Rich has always been there as a colleague and a friend. I also want to mention very briefly that in my first 2 years here, Rich sent me an email and said, look, I've got this HHMI program, and it's about helping students who are here at Harvard who just need a home. They need to find a lab and a mentor they can connect with. People from different walks of life, and I've got just the person for you. And Rich introduced me to an amazing person named Annalise Roquet. She was a freshman. She stayed in my lab for 4 years, an extraordinary scholar. She is now an infectious disease doctor with the CDC, and also bringing her talent and expertise to underserved groups in the United States and abroad. She sends her warmest regards, Rich, from Atlanta, Georgia, and we had a wonderful conversation about all the wonderful things you've done. Now, as Rich is our first Distinguished Achievement Lecturer and a founding member of the Microbial Sciences Initiative, we wanted to give Rich a small token of our appreciation. So I'm going to walk over here, and I have a big reveal. If only we had a drum set. But, oh my God, you all are amazing. Ladies and gentlemen, let's see if I can do this without breaking it. This is our award to Rich. This is a stromatolite, which I did not know Jeff would set us up so well, but this is an ancient microbial fossil produced by photosynthetic cyanobacteria. This is from the Miraflores Formation in Bolivia, and is about 60 million years old. I'm gonna, a little bit. I'm going to riff a bit off of Niels here and say that I too got to thinking, how is this stromatolite like Rich? Well, they're built layer by layer, and the foundations support the growth of others above them. They're the result of different microbial types over time working together, producing something greater than the sum of their parts, and of course, they endure. So Rich, I wanted to thank you for all that you've done. I wanted to see if Mike, you wanted to offer a few words for Rich.
You know, I would echo everything you said, Pete. When I came here in 2004, the opportunity to interact with Rich and work with them and be part of MSI and connect with them was certainly one of the things I was most excited about. I'll say his views and ideas have been really formative for me and many people I work with. He has wonderful insights. You know, the parallels between Avery, McLeod, and McCarthy and Delbruck and Luria are not unlike Darwin. Darwin rediscovered what Adam Smith knew in economics 100 years earlier when he described the silent hand of the market, which was natural selection. It took 100 years later for that to be applied in biology because economists and biologists didn't talk, and because in biology we already knew how life was handed down from the divinity, and we didn't need a new solution. Here we suspected that the transforming principle was probably protein, so we didn't really need a new one. And so it took a while for that to sink in. And so, questioning dogma is something that I think is really the bottom line of that, and a great illustration of the way Rich has approached science and life, and to ask what's real. So with Rich, you've had such an impact on everybody. We all feel very, very fortunate to have you as a colleague, and I think as the first recipient of this, I can't think of anything more appropriate. The last thing I would say is, as you aptly illustrated, the biochemists didn't really speak with the geneticists. And that actually is one of the founding aspects of MSI. People from different parts of microbiology or different that impinge on microbiology didn't necessarily associate, and MSI creates a community where we can. So hopefully we won't be delayed in our discoveries as a result of your founding MSI. So thank you very much for that, and we're so delighted.